Yeah, good morning, everyone. My name is Nerea, and I'm part of the Cambium Research Group in Soria. And today I'm going to give you a presentation about a project that we've been working on, which is about silent vessel segmentation through artificial neural networks. And first, I'm going to give you a little bit of background about what this project is based on and what we do in our group, which is dendrochronology. And we could say that trees are like living history books, since dendrochronology studies environmental conditions that occurred in the past by looking at wood. Trees that grow in temperate climates show seasonality. Trees that grow in temperate climates which have seasonality show growth pulses, depending on factors like temperature, precipitation, light, or even pest attacks. These pulses can be seen in wood as growth rings, which are formed by silent tissue consisting of early wood and late wood. And together they represent a year of growth. To study the growth rings, we can either cut a cross-section of a tree, or we could just take a core sample of a tree, and this way we don't have to kill the tree. So in this core sample, we can see that the growth rings vary in size depending on external factors. And in this case, we see that the ring corresponding to the year 2005, it's quite thinner compared to the other rings. And this is due to a severe drought that occurred in the Iberian Peninsula during that year. And dendrochronology is a great way to study how the environment affects the growth of trees, but it is not the only way. We can actually look closer into the wood, into its xylem tissue and the anatomical structure. The xylem anatomical structure is also influenced by environmental conditions during its growth. And xylem is composed of different types of cells that are responsible for the transport of water and also for giving support to the tree. One of those cells are vessels that are present in angiosperms, but not in gymnosperms. Xylem vessels are specialized in water transport, so they are like tubes, they are hollow. And their diameter can vary depending on climate conditions. So for example, when there's enough water availability, the tree will adapt by growing wider vessels. And the study of the xylem anatomical structure, it's called quantitative wood anatomy, which can help us to study how the environment shapes the growth of trees. However, quantitative wood anatomy requires expertise and time. The sample preparation techniques are highly time-consuming, and they start with a core sample of a tree, which has to be thinly sliced using a microtome, and then these slices are stained and dehydrated to be seen under a microscope, which has a camera mounted onto it, and then this camera can take pictures of the xylem tissue, as we can see here. So to avoid this highly time-consuming process, we have proposed an alternative mechanism that doesn't require microscopy, but just uses digital photography. So we take pictures of tree cores using a device called capturing that can take automatic photographs of cores using a camera mounted on a rack, as you can see there. And then we obtain pictures depending on the camera and lens use that can have enough augmentation and resolution to see the vessels of ring porous species, as you can see there on the right. And so for our study, we use the species Fraxinus angustifolia, the narrow-leaved ash, which is a ring porous species. And what we did was, first of all, take pictures of the cores using capturing. And when we had the cores digitalized, we used a graphical user interface software for, first of all, detect or segment the xylem vessels manually. And then the software will try to replicate this pixel classification and detect the vessels by itself. And sometimes the segmentation goes well, as you can see there, that the vessels are selected, but most of the time the vessels are not correctly segmented because the wood is too light in color, and the software cannot differentiate the vessels from the background. So to also solve this problem, we have proposed another mechanism to segment silent vessels that uses artificial neural networks, specifically convolutional neural networks, which are a type of deep learning algorithm specialized in processing data with grid-like structures, like images. So digital images are composed of pixels, structured in a grid-like fashion, and they consist of red, green, and blue layers that have different pixel values. So knowing this, what we do is to feed a neural network with images of three cores. First, original images without vessel segmentation, and then images with segmented vessels. And what the network does is analyze overlapping sections of these images to extract a summary of features. So then it classifies these features to learn the pattern of pixels that have the vessels. So the network will then output an image with detected vessels that should match the image that we gave it in the first place. And later on, we can give the network more tree cores, and it will automatically segment the vessels. So finally, I would like to say that we're still working on this project, but the conclusions that we can get by now are that we were able to get high-resolution images of tree cores in a short amount of time, and that these images can train a convolutional neural network for automatic silent vessel segmentation. And then we can get a new quantitative wood anatomy method that allows a high throughput of samples to obtain more data about the response of trees to past environmental conditions, which can then help us to estimate how trees will behave under the current climate change scenarios. Thank you very much.